All right, so today I'm going to tell you about burst timing dependent synaptic plasticity and how it can coordinate learning in hierarchical circuits. Um, this is something that those of you who are at Maine would have seen a presentation on from Richard No, but I will give a slightly different light on it as well, and hopefully it'll be new for you, even if you saw his talk. So to begin with, if my thing, there we go. Okay. I'm actually going to do most of this talk in a bottom-up fashion, but I want to give you a sense of where we're ultimately headed, which is a very top-down perspective. So the top-down perspective that I want to give you is this. What does it actually mean to learn? All right. I think that it's important that we get clear on what we actually intend by this term. And I sometimes get frustrated in conversations with people because when we talk about learning, I feel like people have sometimes a funny definition of what learning is in their head. And so I want to get 100% clear on what I mean by learning. So as far as I'm concerned, learning necessitates a normative perspective. And what I mean by that is that unlike other changes that occur to your body or your brain, learning necessarily involves changes that we can identify as being good or bad. Because in order for it to count as learning, the changes that are occurring must be good. They must be achieving some goal. If they're not, it's just changes. So you can look at a system that's evolving constantly, but we don't call it learning unless there's actually some metric that is provably getting better as a result of those changes. And of course, to quantify this normative perspective in machine learning, we use loss functions. And I have argued that neuroscience also needs to adopt loss functions more. For the record, many computational neuroscientists have used loss or cost functions for their work for ages. So it's not that it's new, but what I have been arguing for the last couple of years is that it's a perspective that needs to be taken on not just by a particular subsection of the computational neuroscience field, but instead by the entirety of neuroscience. And it's something that should inform the way that we think about the brain more broadly. Now, if we're measuring learning with a loss function, whatever that loss function is, and we're restricted to relatively small changes to our parameters, okay? Such that, let's assume that we have a smooth landscape and the changes are small enough that the function is close-ish to linear in the scale that we are potentially changing things at. If that is the case, then necessarily, if you get better at something over time, your changes must have been correlated with the direction of the gradient of that loss function. So if I have some synaptic weights at time 0, and then they change to another set of synaptic weights at time t, no matter what path I take to get there, no matter what other changes are being induced by other processes in the brain, if I actually call it learning, this vector of change cannot be completely orthogonal to the gradient of our loss function. It must be at least non-orthogonal. And furthermore, I would venture the following, which is that if you are measuring performance with a singular loss function, and we can talk later about the question of when you have multiple loss functions, which I think is very interesting and relevant to the brain. But if you have a singular loss function, then generally algorithms which adhere more closely to the loss gradient will actually tend to learn faster than those that don't. Now, there are some exceptions to that in terms of obviously you would ideally like things like a second order method, etc. But the direction is still going to be along the direction of your loss gradient. If instead you have an algorithm that provides you an estimate of the loss gradient, but with some variance or bias, learning is going to be slower and not necessarily as good. An example of this comes from this paper from Zi and Sung. Actually, this one might be Werfel and Sung. I apologize. where they compare standard gradient method to node perturbation and weight perturbation. The way that these algorithms work is you induce random changes in your network, and then if the loss function goes down, you keep those changes. If it doesn't, you don't keep them. And this could be implemented in a very simple biological system, and to be honest, I think it's how many neuroscientists think of learning in the brain. But these algorithms are providing you an estimator for the loss function. 1 second, I'll give you a comment in a second. An estimator for the loss function with high variance. And in fact, the variance scales with the size of the networks, such that you are always going to need more data examples to train up a network with node perturbation or weight perturbation than with an actual gradient-based method. Yep. So in fact, what they did is, in this case, they were applying it to very simple networks where they could analytically calculate the scaling with respect to the number. And I forget what the equations are, but they show that basically it gets terrible with node perturbation and weight perturbation as the size of the network. Yeah, that's already too bad. That's already too bad. That's right, exactly. So my argument then, which some people on Twitter consider ridiculous, I don't know, is that until someone demonstrates efficient learning in non-toy tasks without some kind of reasonable estimate of a gradient, it's not crazy to start thinking about potential physiological substrates for gradient calculation in the brain, or some way of getting a hold on the gradient to follow it as closely as possible. Yeah. Yes, sorry. So I agree with that point. What I should say here is that what we want to see is information about the gradient that is relatively low variance and not too biased. Okay, so this is where I'm going. But what I'm going to do is I want to start by telling you a story that's based on biology. And we're going to build up to gradient estimation via a low-level biological model. Yeah. I mean, yes, that's right. Because that's because biologists don't understand what the brain does generally. Sorry, what I mean by that is, I'm joking. I'm a biologist. I have a degree in neuroscience, and I've run many experiments. Yes, that's right. Yeah, I'm channeling Conrad. I'm joking around. Biologists understand a lot about the brain. The reason that they push back against this is because they imagine that this is just us taking whatever the shiny new toy is, deep learning, and saying, oh, let's apply this shiny new toy to the brain. There you go. We're going to solve it all for you, you poor biologists who have been at it for decades. That's right. And it's very frustrating for them. But my point is that this claim that you want to estimate the gradients is actually a fundamental principle about learning. And so it's something that still applies, even though deep learning might be the shiny new bauble in AI. This fundamental idea dates back way before the deep learning era, and I think has always, in fact, applied to the brain. But because I'm aware of the fact that biologists do actually know a fair bit about the brain, what I want to do is I want to build this up from real biology. And so start with a model that's grounded in real biology. Okay, so my talk today is going to describe how this particular plasticity rule that has been observed in the brain, which I'll call burst timing dependent synaptic plasticity, coupled with some of the unique properties of the dendrites of pyramidal neurons, can actually enable hierarchical gradient calculations. Okay, so this is work that has been done by Jordan Gergiev, a PhD student in my lab, Alexandre Payeux, a postdoc in Richard No's lab, Richard himself, and Friedemann Zinke, who's now at Bristol. And this really has been a wonderful collaboration. Richard said that at Maine, and I really do emphasize that. It's one of these unique moments where like everyone's just kind of working together, and it came together so nicely. We should have a preprint up by the end of the month, or early January at the latest. Okay, so burst timing dependent plasticity. To start with, you might have heard of spike timing dependent plasticity, or STDP. I've got some unfortunate news for you in case you like STDP. It's fake news. STDP is not actually a synaptic plasticity rule in the brain. STDP is what you get in a very particular experimental circumstance when you control all these different variables just so. There's a lot of evidence, and I mean a lot of evidence, showing that what really matters for determining whether or not, so for those of you who aren't familiar, sorry, I should say, spike timing dependent plasticity, or STDP, says that the sign of your changes to your synapses should be a function of the timing of pre- and postsynaptic spikes. So if you have the presynaptic cell spike before the postsynaptic cell, you should get LTP. That's long-term potentiation. That's an increase in your synaptic strength. And if you have the opposite, you should get depression, a decrease in your synaptic strength. But in fact, what the biological evidence is very clear on is that the thing that determines the sign of change in your synapses in real neurons, and I can say this as someone who's run these experiments as well, like definitely the easiest thing that will determine what change you get in your synapses is how depolarized your postsynaptic cell is. That is how active your postsynaptic cell is. And in particular, what matters is the depolarization in the dendrites, which are the site of synaptic integration. Now, it turns out that the best way to depolarize your dendrites is with what we call a high-frequency burst. That is a set of multiple spikes that occur in a high-frequency train in a row. And what you can show in experiments, such as these ones here, so this is at the bottom, this is the synaptic strength. That stands for excitatory postsynaptic potential amplitude. So this is basically a measurement of synaptic strength for one neuron that connects to another. And they've patched on with electrodes here to measure the synaptic strength and manipulate the activity of these neurons. And what they do is they ran, this is a study from Letskis et al. From 2006. They ran a protocol where they caused the postsynaptic cell, illustrated with the blue electrode here, to spike just after the black cell here. So they cause the presynaptic cell to spike, and then they get the postsynaptic cell to spike. And if you do this at a low frequency, so you go bup, bup, wait a few seconds, bup, bup, wait a few seconds, you don't actually get an increase in the synaptic strength, which is what spike timing dependent plasticity says you should get. Instead, you actually get a depression in the synaptic strength, measured here. So that's this protocol here. In contrast, if you now depolarize the dendrites of this cell in order to elicit something called the plateau potential, which I'll tell you a little bit more about in a second, and this induces a high-frequency burst, you convert that decrease in your synaptic strength to an increase in your synaptic strength. And the reason is because that burst really depolarizes the dendrites. And that depolarization is the key biological signal that then unleashes a biochemical cascade that leads to an increase in the synaptic strength. So let's model this, basically, is what Richard and I said. Yeah? So the reason the apical dendrite matters, and I'll come back to this, is because the apical dendrite is what determines whether or not you get a burst. But the same principle applies in any dendrite. The plasticity is going to be determined by the degree of depolarization. If you get just a little bit of depolarization, you get depression of the synapses, and if you get a lot of depolarization, you get potentiation of the synapses. You don't even need spikes. In fact, this has been something that has been proven multiple times over. If you just directly depolarize the dendrite and no spike occurs, you get potentiation if it's high enough. And if you do it just a bit, you get depression. Because that is the biological signal.
There's an expected amount?
Yes.
Your blood is good?
That's right. Yes, exactly. Yep. The only thing that the presynaptic cell matters for is it must have been activated to see any change occur. So people have shown that if you don't have synaptic inputs and you just depolarize the dendrite, you're not going to get any changes. It has to be that the depolarization is paired with a presynaptic input. And we think that's because the other key biological signal is basically the cascade that emerges from the neurotransmitter being received at the site. There's effectively two things that need to happen to get plasticity by virtue of something called the NMDA receptor. You need some degree of depolarization at the postsynaptic site and some presynaptic inputs. And that will then give you plasticity. Yep. Yes, that's right. Sprinkle on some glutamate, depolarize the dendrite, boom, you got plasticity.
So stabilized.
Yes.
Are above, then the weight increases.
Potentially, that's right. So let's come to this plasticity rule. So the way we model this is relatively simple. The idea is that if you have a presynaptic input, whether it's a burst of spikes or a single spike, the thing that determines whether you engage in long-term potentiation, that's an increase in the synapse, or long-term depression, decrease in the synapse, is whether or not the postsynaptic cell is bursting. So if the postsynaptic cell is bursting when you get presynaptic inputs, or just after you get presynaptic inputs, you get potentiation. If it's not, and you just have an individual spike, you get depression. And we model that with this equation. So these are, I should say at the start here, everything I'm going to show you is biophysical models. We are explicitly modeling membrane fluctuations and with all the leaky channels and stuff. And we also have like continuous time simulations of the synaptic weights. So what's happening is the synaptic weights are updated according to this rule. Their change is determined by, of course, a learning rate, but that's just a hyperparameter for us. And these variables here. So critically, BI here is an indication of whether or not a postsynaptic burst occurred around time t. So when a burst occurs, you get this Boolean variable saying, yes, a burst has occurred. Yep. A burst is a high-frequency train of spikes. So, well, that in this rule, you need spikes because we're not modeling down to the level of an individual synapse in the dendrite. So we are abstracting out. The thing is, in vivo, typically what's going to drive depolarization is spikes and bursts. Now, it doesn't have to be, and there's some situations where that's not the case, but that's a good question. Here in this rule, yes, you're going to need a burst to get a change. We're assuming that this is the source of depolarization. But what we're doing is we're not just taking whether or not you get a burst. We're comparing that. We're scaling this effect with a running average of the burst probability, and this gets to your point. So there is this stabilization effect whereby basically as you get more and more bursts over time, the degree of potentiation that you get decreases. Yep. Yes. It's not too different from it, but there will be situations in which the learning rules diverge because triplet STDP rules still have very complicated predictions about specific spike trains that don't necessarily match this one. But you're right, they're not radically different in flavor. EI here is just a measurement of whether or not a postsynaptic spike or burst occurred. And the reason this gets included now is let's say the postsynaptic cell just spikes and it doesn't burst. What you now have is a negative term here. So you get depression of your synapse. And then this is a presynaptic eligibility trace, which is basically a sort of exponential kernel that we convolve with the spike trains in order to determine when presynaptic inputs have arrived in the recent past. Now, yep. Sure. So this is a Hebbian learning rule in some ways. It depends on what you classify as a Hebbian learning rule. So the term Hebbian learning has both a broad and a narrow interpretation. The broad interpretation is, yeah, just presynaptic activity and postsynaptic activity together determine potentiation or depression. The much more narrow idea is fire together, wire together, which says just that if there's correlated activity between your pre and postsynaptic cell, you increase the strength, regardless of the nature of that correlation. Here, even if you have correlated spikes, you're going to get depression. You're not going to wire together. It has to be that the presynaptic inputs are correlated with postsynaptic bursts. Now, one of the things that we get out of this, so when I said to you STDP was fake news, an example of where it is fake is, so STDP says that if you have a presynaptic spike followed by a postsynaptic spike, you should get potentiation. But the reality is that experimentally you can see that that only actually happens if you do those pairings at a very high frequency. So if you do pre-post at like 100 hertz, you get potentiation. But if you do it at 5 hertz, you actually get depression. That's been shown experimentally. And indeed, that's what happens with our learning rule. The reason is that as you increase the frequency, your train starts to become a burst. And so it converts into potentiation from depression. We can replicate this basic experimental result that was originally shown by Jesper Sjöström, who's a researcher here at McGill now, and has been verified by many other people. So that was nice to see. The other thing that's interesting is that this produces something that looks a lot like the Bienenstock-Cooper-Monroe rule, for those of you who are familiar with that. That's a rate-based rule, which says effectively that the degree of potentiation or depression should be determined by your postsynaptic rate. And the other thing it has is a sliding threshold for where you engage in potentiation or depression based upon your recent history. And BCM has been shown to replicate a variety of biological features in terms of receptive field tuning and stuff like that. And lo and behold, what we actually get out of running a protocol where we do Poisson spiking at different rates is the Bienenstock-Cooper-Monroe rule. It just emerges out of the plasticity rule. Okay, so that's cool, and like by itself, that would be a kind of fun computational neuroscience study. But as you know, I'm interested in gradient calculations. So how are we going to get from here to gradient calculations? And the way we're going to get there is courtesy of the apical dendrites. Okay, so I mentioned earlier that the apical dendrites have special properties that are relevant to this question. So for those of you who are unfamiliar with them, pyramidal neurons are the most common type of neuron in the neocortex, which is the region of the brain that's concerned with kind of higher order functioning. And neocortical pyramidal neurons have this interesting structure where their cell body is surrounded by one set of dendrites called the basal dendrites. And then they have this big, long branch that they send off called the apical dendrite that heads off towards the surface of the brain. And then there's another set of dendrites called the apical tuft up towards the surface of the brain. And what's interesting is that anatomical work has shown that these two different sets of dendrites receive different inputs to a large degree. Now, it's biology, so it's never 100%, and let me just clarify that. But what we can say is that largely the inputs to the basal dendrites are coming from lower level areas. So they're going to be carrying information about what light is hitting your retina, what's touching your skin, what sounds you're hearing, etc. In contrast, top-down feedback from your higher order association areas, which are, where you're forming your larger understanding of the world and potentially your expectations for what you should be seeing, those come into the apical tuft. And interestingly, the relationship between these apical dendrites and the soma is not what you'd typically think of. So Normally, we think of dendrites receiving inputs and propagating it through to the soma in a passive manner. But that apical trunk is so long, it's electrotonically very segregated from the soma, such that when you get an input to the apical dendrites, that input is not actually propagated down to the soma unless there's a nonlinear event that occurs, which I'll tell you about in a second. For the passive propagation, that's shown here. So these are these experiments done 2 decades ago now by Matthew Larkham, and they're really beautiful experiments. I cite this paper all the time. What he did is he patched the same pyramidal neuron in 3 different locations, color-coded here according to the electrodes. And he would give an input to the red electrode here, and then look at how it came out at these other 2 electrodes. And what you can see is if he just does a passive propagation, where he just gives a little bit of input to the red electrode, you can see it on the red electrode, you see it a little bit on the blue electrode, and by the time you're down at the cell body, which is where spikes are generated, you see nothing. So inputs from the apical dendrite, when they're passively propagated, literally cannot reach the soma. But the way in which the apical tuft can communicate with the soma is by virtue of a set of nonlinear calcium channels that sit in this trunk. And these nonlinear channels allow the apical dendrite to engage in its own nonlinear signaling, not too much different from a spike, though different in its temporal dynamics, which we call a plateau potential. The way this works is that when you get sufficient depolarization of the apical dendrite such that it hits a threshold, basically, the calcium channels all open up, and when they open up, positive current flows into the apical dendrite, and they remain open for a certain period of time, and this gives you this big depolarization of the dendrite, seen in red here, which lasts for quite a long time by virtue of the calcium channel dynamics. We call this a plateau potential because it lasts for a long time. And that plateau potential is propagated down to the soma. In fact, it has such a big impact on the soma that it typically drives bursts of high-frequency spikes. So these plateau potentials are, in fact, how you get bursts. And the way you can think of this is that they are, in fact, a coincidence detector. So what happens is, this is a biophysical simulation done by Adam Shai. Here we have a fully multi-compartment simulation of a pyramidal neuron, and he's got synaptic inputs to the apical tuft and synaptic inputs to the basal dendrites. And what happens is he shows that if he drives inputs to the basal dendrites, he gets a single spike out of the cell. If he drives inputs to the apical tuft, what's going on there?
Bursting.
It's bursting, I know. If he drives inputs to the apical tuft, no spike occurs because it can't propagate passively. But if the apical tuft and the basal dendrites are activated simultaneously, you can hit threshold in the apical branch. That causes a plateau potential, which generates your high-frequency burst. So the result is that inputs to the basal dendrites will drive single spikes. Inputs to the apical dendrites do nothing by themselves, but if you get a pairing of basal inputs with apical inputs, you get a high-frequency burst of spikes. Now, what this means for our plasticity rule is that now the inputs to the apical dendrites become a teaching signal, effectively, because they're going to determine whether or not your cell is bursting. So if you have 2 pyramidal neurons connected where one pyramidal neuron connects to the basal dendrites or the perisomatic region of the other, and then you have some distal inputs to the apical dendrite here, those inputs are going to determine the sign of plasticity at this synapse. So these are biophysical simulations again. So we've got a multi-compartment model with voltages fluctuating and all measured or simulated in continuous time. And what happens is, here we're showing the probability of a burst in this neuron. And as we increase input to the apical dendrite, which is in the red here, we can see an increase in the postsynaptic burst probability. sorry, that is the postsynaptic burst probability, which is resulting from us increasing inputs to the distal dendrite. If we decrease the distal inputs, we decrease the probability of getting a burst. The result of these changes are, if we measure them at the weights, increases in the synaptic weights when we increase the inputs to the apical dendrite, and decreases in the synaptic weight when we decrease the inputs to the apical dendrite. So you've got this ability with your apical dendrite to control whether or not you're potentiating or depressing. Yep. In that, yes, it's on the apical trunk in that paper. So no one's actually run these experiments before. This is a prediction of ours that we're trying to convince someone to run the experiments on. Yeah. Yes, like I'm saying, I want someone to do those experiments. Very much so. They're tough experiments. Like, the way to actually do this properly would require a lot of finicky things. So it requires someone committing, a PhD student a year of their life, basically, to running these things. So it might take a while to convince someone, but I hope to. But then they get famous, yes. If it works, that's the problem. If they don't believe me, then they're not going to try. So the experiment is, you do a plasticity protocol from one pyramidal neuron to another, where you know the synapse is on the basal dendrites, and then you simultaneously inject current into the apical dendrites, and you should be able to show that you can determine whether you get potentiation or depression by controlling the apical dendrite. This has been done with multiple inputs to the apical dendrite, and they've shown this. And the reason they do that is because it's slightly easier, because then you've already got your patch on the apical dendrite, so the thing you're injecting current with is also what you can measure the synaptic input. Doing this experiment would require like multiple patches on the same cell and a plasticity protocol. It's difficult, but doable. Okay. So the other thing that this function of the apical dendrites does is it gives you the ability to multiplex top-down and bottom-up inputs. So this was something shown by Richard, it was how this collaboration started. If you simulate an ensemble of pyramidal neurons that have these properties with biophysical properties fit to experimental data, and you inject current into the perisomatic region, shown in blue here, or the distal apical dendrites, shown in red here, you can show that what you end up getting out are two different signals out of the same spike train. Now, the way you can measure that is by differentiating between events and bursts. And I've already kind of mentioned this, but the idea is that we define an event as either a spike or a burst. And then for any event, you can ask, was it a burst? So we can talk about, across the ensemble, the event rate. That is, how many of the pyramidal neurons in the ensemble are engaged in an event right now. Simultaneously, you can ask, what is the probability of bursting across the ensemble? And that's a function of how many of the events are bursts. So this gives you two dissociable signals, shown here. So as we increase the somatic input, we can see here we only get one event out of across these three. So these are the voltages of three of the simulated pyramidal neurons, 1, 2, 3. Only one of the pyramidal neurons here gave an event. So our event rate is low. But that event was a burst. So the burst probability is high. We can get an increase in the event rate such that here we have all three cells giving an event. So we have a high event rate at the same time as we get a decrease in the burst probability, because fewer of the events are bursts. So these are two dissociable signals that you can read out of the population. And what Richard showed was that if you look at the inputs to the apical dendrite and the somatic region, if you just measure the firing rate of a neuron, it's hard to see any sign of those inputs. It's just a bunch of noise, basically. But if you measure the event rate, you see the inputs to the somatic region. And if you measure the burst probability, you see the inputs to the apical dendrite. So this means that a group of neurons can be simultaneously broadcasting both what they're getting to their basal dendrites and what they're getting to their apical dendrites. This then means that you can, in fact, propagate top-down and bottom-up signals simultaneously. So it requires a little bit more machinery involving some inhibitory interneurons, and it also requires a particular assumption, which we view as another experimental prediction from this model. Okay. Which is that the bottom-up synapses, we did start late though, but oh, you got that. Okay, very good. No, you didn't. It's one anyway. Oh, but you're giving me talk time. Yeah. Okay. Very good. So, what we have here is we're assuming that the synapses in the basal compartments and the inhibitory neurons that connect to the basal compartments are driven by what we call short-term depressing synapses. These are synapses that after they've received one input show a decrease in the response to subsequent inputs. And this is something we actually know from biology that these synapses are short-term depressing. But there's another set of synapses that you can see in the biological literature, which are called short-term facilitating synapses. And these are synapses where when you get an input, it's actually very weak, and you only get a strong response to that input if it's followed by subsequent inputs very rapidly. And the difference between these things is that these synapses are going to be totally indifferent to bursts, whereas these synapses, the short-term facilitating ones, will only care about bursts. So if you assume that your bottom-up and top-down synapses have these different properties, and as I said, we know these ones do. Yeah.
Is there evidence for that?
So we know these synapses have short-term depression at their sites. And in fact, we also know that the inhibitory interneurons here have short-term facilitating synapses. So 3 out of 4 of these synapses have been demonstrated experimentally. The fourth one, the top-down inputs, have not been studied. So it's an explicit experimental prediction that top-down inputs should have short-term facilitating synapses. So it's a bit complicated to explain here, but basically the interneurons are how you end up dividing things out to get the signals that you're actually interested in with the 2 different pathways. I can show you later.
It's a numbers problem.
You can have very few interneurons. It doesn't have to be a one-to-one matching. This can all be driven by just a single interneuron. It's just about controlling the homeostatic level of the voltage. Yeah, that's right. That's right. It's just normalization, effectively. If you do this, what you get is that when you have one set of pyramidal neurons receiving basal inputs and another set of pyramidal neurons receiving apical inputs, so here we can see the apical inputs are being driven by a slowly oscillating current. The basal inputs down here are being driven by a rapidly oscillating current. That rapidly oscillating signal will be reflected both in the event rate of these neurons and the event rate of these neurons. So that bottom-up signal gets propagated upward to the next set of neurons. Simultaneously, the burst probability in these neurons it's also going to fluctuate with the faster oscillation, but it will be amplitude modulated by the slower oscillation coming from down here. And so what that means is you now have a signal that is communicating this burst probability from the top, which is dependent on the current you're injecting into your apical dendrite, get communicated down to these neurons at the same time as they're communicating their event rate forward. So now you can have these groups of neurons communicating bottom-up and top-down signals simultaneously with each other. If we combine these two facts, you can actually do hierarchical credit assignment using BTDP in pyramidal ensembles. Continuing with the biophysical simulation for a moment, we do this very simple simulation because these biophysical neurons, like to clarify, you're all familiar with heavy compute needs, but trust me, you don't know heavy compute until you've tried to simulate a biophysical neuron or thousands of biophysical neurons. It's a nightmare compute-wise. So we keep this very simple. We have groups of pyramidal neurons arranged in ensembles like this with an input layer, a hidden layer, and an output layer. And just what we're going to do now is we're going to train it on XOR. So we're going to give inputs to the basal dendritic regions of the 2 inputs that match 0, 0, 1, 0, 0, 1, or 1, 1. And then we're going to train these things by providing a teacher signal to the apical dendrites, where we're going to activate the apical dendrites when we have 1, 0, or 0, 1, and not when we have 1, 1, or 0, 0. This is what it looks like. So before training, this is the output that we get from the output neurons. At first, it basically just gives low output when there's nothing, and then higher output when there's something. And after training, it's now learned to step up its output. Oh, its overall output is higher, I should say. There are biophysical reasons for that. But it has now learned to step up its output to the 1, 0, or 0, 1 cases and step it back down to the same level as the 0, 0 case when it gets 1, 1. And this was all just by running this in continuous time and providing these steps of current to the apical dendrites here at the end of each epoch in which we're showing, or period of time in which we're showing the pattern to the inputs. These are just the error signals, but in the interest of time, I'm going to skip through this a bit. Okay, yeah, sure. Yes.
Inputs are plateaus.
That's right.
What's the relationship with the time scale?
Yeah, so it is important that you give sufficient time for the activity to propagate through the circuit, and then you need to also give a pulse. The length of the pulse doesn't actually matter a super amount, as long as there's long enough for it to get to the hidden units here. The larger this network is, the more time you'd have to give for the teacher pulse so that it could propagate down. It is timing sensitive, yes. Okay. So to actually get a hold on gradients, what we have to do is we have to zoom out to the ensemble level. So the idea is that now we're not going to simulate individual biophysical neurons. We're going to instead treat each ensemble of pyramidal neurons as a unit unto itself. And we're going to treat those units as having two different values, an event rate and a burst rate. The event rate is just a standard neural network type input. It's sigmoid applied to linear function. We then have the burst probability determined by, this is the feedback of the burst rate from the higher layer. That's layer I plus 1. So I here indicates layers, and these are vectors for the record. So this is the burst probability from the upper layer times a feedback synapses. And then we also need some homeostatic control over these dendrites, and I'll explain why in a second. That's done with this negative z of the burst rate of this layer here. And our burst rate is then just the probability of bursting times the event rate. So these are our units. And what you can show is that if you take the limit of an infinite size ensemble, which is obviously not how biology works, but it makes the math easier. If you take the limit of an infinite size ensemble, you get out a learning rule that looks like this. This is what burst time independent plasticity turns into in the infinite limit. And what it turns into is the difference in the current burst probability minus a running average of the burst probability, and then a Hebbian multiplication of the event rates of the presynaptic, so that's the presynaptic and postsynaptic neurons. And that's our weight matrix update. So there's our current burst probability, moving average, and then our two event rates, as I said. And you can show that if you make two assumptions. Now, Joshua also asked another question at Maine, which I haven't addressed here, which is there's also some considerations about the nature of your activation function here, which I'm happy to talk about more in questions. But the two key assumptions are this. If you assume that your feed forward and feedback weights are symmetric, and that your burst probabilities are in a linear regime, then you can show that this is gradient descent on a mean squared error loss function, or a loss function where you're receiving apical inputs at the top of your hierarchy. Now, this is now why we use this feedback pathway. That's to get the linearity of our burst probabilities. Yeah? this learning rule is what that learning rule turns into when you take an infinite ensemble of pyramidal neurons. The limit of the other one gives this. Now, so what we do is we use this pathway because the burst probabilities have to be between 0 and 1. So we're calculating those with a sigmoid function. And in order to actually be following the gradient, we need that sigmoid function to be sitting on the linear part of the curve. So we control that with a set of inhibitory interneurons that are learning to regulate the apical dendrites homeostatically and keep them in a kind of midpoint range. And we can show that if we do that, this is the difference between our algorithm, the angle between our algorithm and regular backprop in a network trained on MNIST. If you don't train these weights, you still get some alignment because of something called feedback alignment, which is a whole other talk, so I'm afraid I'm going to have to leave that for now. But we can crunch down the difference with the gradient even more by linearizing those burst probabilities, shown in the red curve there. Additionally, you need to get feedback weights that are symmetric. Now, it just so happens that there's a simple algorithm for this called the ColinPollock algorithm, which we can adapt easily here because what ColinPollock basically just says is that you update your feedback weights with the same update as the feed-forward weights plus some weight decay. And because we are updating the things just with bursts, which are available at synaptic sites, that's easy to postulate. So we literally just update the feedback pathway with the same updates minus some weight decay. And what you get then is symmetric weights. And the result is that you're following the gradient pretty well. So here's what you get if you train one of our networks with just a feedback alignment effect, shown in the green trace. But if we add this ColinPollock stuff, and this is our burst timing dependent plasticity rule, infinite limit rule that we're training with, then you follow the gradient quite well. And on something like CFAR10, you can crunch it down to the same level you get with backpropagation. And we've been running this with a whole variety of other data sets, and it looks like we can always match backpropagation on any data set. Yeah, so what it would require is that the feedback synapses are also changing according to the difference between the burst probability and a running average of the burst probability. You need the sign of the feedback changes to equal the sign of the forward changes. Yeah, so we can. So what it requires is that you're also paying attention. That's right, to the postsynaptic events, and you need the presynaptic inputs. So the assumption is that the cell knows what the other synapses are doing in it. Not crazy. It's assuming that their biological signals are shared throughout the cell, because it doesn't have to be communicated across cells critically. Okay, I should finish up. This is the end anyway. So, in general, yeah, I know. Some species, especially humans arguably, can learn many things very efficiently. And I think one argument that I always find amazing, so I have discussions with biologists who I respect immensely and who are incredibly smart people, like Tony Zador, if you saw him speak at Maine a while back, or a couple of weeks ago. Tony's a brilliant guy, and he makes really cogent arguments about innate behaviors, and he knows a lot about the brain and all of these innate behaviors that exist. And he's right that there are tons of innate behaviors. But then he makes this following leap. He says, okay, there's tons of innate behaviors, and so therefore I don't think learning actually is something that matters. And it's that step that I don't get. Because even if you have lots of innate behaviors that have been given to you by evolution, any species that also developed on top of those innate behaviors sophisticated learning mechanisms would totally outcompete the species that didn't have those sophisticated learning mechanisms. So arguably, I think it's not unreasonable to think that evolution has endowed, especially generalist mammals like us, with pretty good learning algorithms that can learn very efficiently. And part of that, as I articulated at the beginning, is to get some way of estimating your loss gradients. That's going to give you efficient learning. And what our work does is it provides a proof of principle as to how you can start with a biological learning rule. And if you then just add a few trappings to it, you can show good estimation of gradients. Now, is the brain actually doing exactly this algorithm? I have no idea. That's something to be determined by experimental testing. We've already discussed some of the predictions that come out of this model, such as the short-term facilitating synapses at the top-down feedback, the way in which the apical dendrite would control the plasticity of the basal dendrites. And another example of a prediction that our model makes is that the variance in burst probability should correlate positively with the error that the individual is making on some task. So what we would predict specifically is that if you drop some electrodes into the brain of an animal learning some task, when it makes errors, you should see increases not in the burst probability, but in the variance of burst probability across the cortex. These are the sorts of things we will hopefully convince experimentalists to actually test for us. So ultimately, what I argue is that we should be doing top-down and bottom-up simultaneously, both in the brain and in the field. And that's that. Thanks very much for listening.